Thanks for the introduction. Thanks to Sofiane for inviting me, and thanks Annette Dolphin for suggesting my name. So today I'm going to tell you about the tools that we designed to selectively detect specific NMDA receptor subtypes in the brain. So the capacity of the brain to interact and adapt to its environment depends on the dynamics of chemical synapses. At the synapses, a neurotransmitter is released from the presynaptic neurons and acts on postsynaptic neurotransmitter receptors. And in the mammalian brain, glutamate is the major excitatory neurotransmitter and acts on postsynaptic ionotropic glutamate receptors, among which NMDA receptors play a key role in synaptic transmission and plasticity. This is the process that underlines learning and memory due to their high permeability to calcium. NMDA receptors are also targets of strong therapeutic interest because their dysfunction is involved in a lot of neurological and psychiatric diseases like depression, schizophrenia, autism, Alzheimer's or Parkinson's disease. At the molecular level, NMDA receptors are tetramers composed of 2 GluN1 subunits that bind glycine and 2 GluN2 subunits that bind glutamate, each subunit having a similar architecture with a large extracellular region composed of an N-terminal domain that is involved in receptor assembly and allosteric modulation in the case of NMDA receptors, an agonist binding domain that binds the agonist, so glycine or glutamate, a transmembrane domain that contributes to the ion channel, and an intracellular C-terminal domain that is involved in receptor trafficking and intracellular signaling. So while the GluN1 subunit is encoded by only one gene, there are 4 types of GluN2 subunits, GluN2A, 2B, 2C, and 2D, that confer to the receptor distinct biophysical and pharmacological properties, as well as various signaling and expression profiles. This subunit can integrate into what we call diheteromeric NMDA receptors that contain twice the same GluN2 subunits, or triheteromeric NMDA receptors that contain 2 different GluN2 subunits, giving rise to a large functional diversity of NMDA receptors in the brain. And while large transcriptomic data have given us a good picture of the spatial temporal distribution of the subunits in the brain, how these subunits incorporate into the different complexes remains challenging to investigate. For instance, in the adult forebrain, the 2 major GluN2 subunits are GluN2A and GluN2B, but they can associate either into diheteromeric GluN1, GluN2A, and GluN1, GluN2B NMDA receptors, or triheteromeric GluN1, GluN2A, GluN2B NMDA receptors. And so the question that we ask is what is the relative proportion of these different subtypes in different brain regions, and what are the pathophysiological roles of each subtype? I mean, can we attribute one subtype to a specific physiology or a pathological role? So currently, there is a large controversy around the specific roles of GluN2A and GluN2B containing NMDA receptors in the brain. So GluN2A containing NMDA receptors are thought to be mostly synaptic, meaning expressed at the synapse. They are thought to be mostly expressed in long-term potentiation, meaning the long-term increase in the synaptic strength, and they are thought to be mostly linked to pro-survival pathways. On the contrary, GluN2B containing NMDA receptors are thought to be mostly extrasynaptic, so outside of the synapse, involved in long-term depression, so long-term decrease of synaptic strength, and they are thought to be the subtype responsible for neuronal death upon overactivation. But this dichotomy is largely controversial in the field, and part of this controversy probably stems from the presence of these triheteromers, for which we have no selective tool to selectively control them. Indeed, pharmacological and genetic approaches are limited to selectively target one single subtype. For instance, pharmacological agents aiming at decreasing the activity of GluN2B containing NMDA receptors, so the diheteromers, will also partially affect the triheteromers. And similarly, genetic approaches aiming at, say, knocking down the gluN2B subunit, will suppress both the diheteromers and the triheteromers. So we need better tools with enhanced molecular specificity. So optogenetic pharmacology allows reaching such high level of molecular selectivity. So this approach is based on the covalent attachment onto a genetically modified receptor, so usually a cysteine mutated receptor, of a photoswitchable compound that can alternate between an inactive configuration and an active configuration upon shining of a specific wavelength, and back to the inactive configuration upon shining of a different wavelength, hence allowing reversible control of the receptor with light. And thanks to this approach, we can achieve strong spatiotemporal resolution thanks to the use of light, cell type specificity thanks to the use of genetics, and strong molecular specificity because the ligand is directly attached to the subunit of interest. So we decided to design such tool to target gluN2B containing NMDA receptors. So for this, we chose gluN2B, an allosteric site, which is the polyamine site at the interface between the N-terminal domains of the gluN1 and the gluN2B subunit. And these polyamines, like spermine, selectively potentiate gluN1, gluN2B diheteromers while leaving the other diheteromers almost unaffected. So we designed a photoswitchable cysteine reactive spermine derivative that contains a maleimide, which is the cysteine reactive group, an azobenzene, which can alternate between a trans extended configuration that is stable in the dark and a cis-band configuration upon shining of UV light, and back to trans upon shining of blue-green light. And the idea would be to attach this derivative called MASP onto a cysteine close to the polyamine binding site, so that in one configuration, the compound is outside of the binding site and so leaves the receptor in its basal state. And in other configuration, the spermine moiety reaches its binding site and increases the activity of gluN1, gluN2B, NMDA receptors. So for this, we screened a lot of attachment position of MASP onto the gluN2B subunit. So we mutated one by one all the residues of the polyamine binding site on the gluN2B subunit into a cysteine. So we co-express the cysteine mutated subunits together with gluN1 in xenopus oocytes to have membrane expression of the mutated receptors that we label with MASP, and then we recorded by 2 electrode voltage clamp the NMDA currents upon different illumination wavelengths. And what we observed is that for one position, R187C, we could, if you apply glutamate and glycine, we have a current that is mediated by NMDA receptor. We could observe a 4-fold potentiation of this current that was fully reversible, with UV, that was fully reversible upon shining of green light, and this process was fully reproducible for many cycles of illumination. Importantly, MASP labeling of xenopus oocytes expressing wild-type gluN1, gluN2B receptor did not elicit any photodependence of the NMDA currents, showing the specificity of the MASP labeling to the attachment of this cysteine. So in the rest of the talk, I will call this mutated receptor labeled with MASP the Opto2B tool. So what is the selectivity of the Opto2B tool? So we tested the effect of MASP labeling on the different NMDA receptor subtypes. So this is what I showed you before. On the gluN1, gluN2B diheteromers containing the cysteine mutation, we have a strong photomodulation of the currents, but almost no effect on wild-type gluN1, gluN2A receptors, and similarly no effect on 2C and 2D NMDA receptors. And interestingly, if the gluN2B subunit with the cysteine mutation is expressed in the context of a triheteromer, so together with the gluN2A subunit, so we have a way in the lab to constrain the stoichiometry of NMDA receptors, we do not observe the strong photomodulation of the diheteromer. So this tool is exclusively selective for one single subtype, which are 2B2B diheteromers. So receptors that contain two gluN2B subunits, and this is actually the first tool that can discriminate between these 2B2B diheteromers and the triheteromers. So now that we have this very specific tool, we wanted to implement it in vivo. So for this, we designed a knock-in mouse, the Opto2B mouse, for which the gluN2B subunit endogenously has the cysteine mutation at position R187C. So we made acute brain slices from these mice that we labeled with MASP, and then we recorded the NMDA current by electrophysiology, so whole cell patch clamp, in the hippocampus. So we recorded current from CA1 pyramidal cells upon stimulation of the CA3-CA1 synapse, which is the prototypical synapse to study NMDA receptor physiology. And this under different alternating wavelengths of illumination. So we first look at NMDA postsynaptic currents, so EPSCs. And on slices from Opto2B mice labeled with MASP, we observe a stronger NMDA EPSCs in presence of UV light compared to green light. And this increase in NMDA EPSCs could be relieved by reshining green light and reset by shining UV light and so on. So we could toggle on and off NMDA receptor activity as we observed in vitro. Importantly, slices from wild-type mice labeled with MASP did not show this UV increase in NMDA EPSC amplitude, showing that MASP has no off-target effects on other NMDA receptor subtypes. So this increase here, we know that it's mediated by the increase in activity of 2B2B diheteromers. So we used the ratio of current under UV light over the current under green light as a proxy for the proportion of 2B2B diheteromers at the synapse. And we looked at this proportion during postnatal development, so from P5 to P40, and we observed the decrease of the photomodulation ratio over development, showing a decrease in the proportion of 2B2B diheteromers at the synapse with age. So what is the origin of this decrease? So it is known that at early postnatal ages, the major gluN2 subunit is the gluN2B subunit, and as the mouse ages, this gluN2B subunit gets restricted to the forebrain where it stays at a high level. On the contrary, the gluN2A subunit is not expressed before birth. Its expression starts right after birth and gradually increases in the brain. So that in the hippocampus, so at the beginning we have only gluN2B in pyramidal cells, and at the end we have a mixture between gluN2A and gluN2B. So we wanted to test if the decrease that we observed was indeed due to the gradual incorporation of gluN2A containing NMDA receptors at the synapse. And for this, we crossed our knock-in mouse with gluN2A knockout mice to suppress expression of the gluN2A subunit. And in these mice, they are supposed to express only 2B2B diheteromers, and we expect, if our hypothesis is true, to see an increase in the photomodulation ratio. And this is the case. In gluN2A Opto2B knockout mice, we saw at every developmental stage a stronger photomodulation ratio compared to the Opto2B mice, showing that gluN2A containing NMDA receptors already populate synaptic sites at early postnatal ages, so before 2 weeks of age, and that this decrease that we observe is indeed due to the incorporation of gluN2A containing NMDA receptors during development. So we also looked at tonic currents, which is the currents that we observe in neurons without doing any stimulation. So part of this current is mediated by NMDA receptors that are activated by very small amount of tonic glutamates in the extracellular medium. And they can be uncovered by the fact that APV, which is a selective NMDA antagonist, inhibits part of this tonic current. So if we, and this tonic current reflects extrasynaptic NMDA receptors. In Optochemical mice, we observe a strong and transient increase in the tonic current upon shining UV light that was abolished with APV and that was not present in wild-type mice, showing that this increase in tonic current reflects the potentiation of 2B2B diheteromers. So we used this increase as a proxy to quantify the amount of 2B2B diheteromers at extrasynaptic sites. And so like for synaptic currents, we observe a strong decrease of the photomodulation ratio between P5 and P40. At P5, we have a fourfold photomodulation, so that was almost the rate that we got in vitro for a pure population of 2B2B diheteromers, and it dropped to 10% photomodulation at adult stage. So it shows a decrease of the 2B2B contribution with age. We were quite surprised because in the literature it is said that 2B2B diheteromers remain at high level and a high proportion at extrasynaptic sites even during adulthood. So we tested if this decrease was also mediated by the incorporation of gluN2A containing NMDA receptors. So we did the same experiments in Auto2B gluN2A knockout mice, and in these mice we observed a strong photomodulation at every age. So showing that this decrease is indeed due to the incorporation of gluN2A containing NMDA receptors, like for synaptic sites. And that was pretty new compared to what was said in the literature before. So in conclusion, we have developed a tool that can discriminate between 2B2B diheteromers and 2A2B triheteromers. And by allowing us to directly look at one single subtype, we could determine the subcellular distribution of these diheteromers in the brain, and also almost solve like 20 years of controversy about the repartition of these subtypes between synaptic and extrasynaptic sites. So we show that at early postnatal ages, extrasynaptic site is almost exclusively composed of 2B2B diheteromers, while synaptic sites already incorporate gluN2A containing NMDA receptors. So it can be 2A2A diheteromers or 2A2B triheteromers. We cannot discriminate between those two. And in adulthood, the 2B2B diheteromer population is minority, and both synaptic and extrasynaptic sites mostly incorporate gluN2A containing NMDA receptors. So with this, I'd like to thank the team of Pierre Paoletti, in which I am a permanent researcher. So this, since we are dealing with anniversary, I actually started working on this project in 2015 when I went back from my postdoc. So it's been a 10-year work. And right now it's in bioRxiv. If you want to scan this QR code, you can access to the full study, and it's going to be published soon in the EMBO journal. So it has involved a lot of M2 and PhD students, in particularly Zakaria Mostefai, Antoine Sicard, Sophie Shi, who's taking over the project now. A lot of collaboration between chemists and neurobiologists. And I would like also to advertise the meeting of Canoionic that will take place in SET, in which I am a co-organizer. So the registration deadline is soon, so please hurry and please register. It's a very nice setting, especially for trainees. And this is where we take our breakfast, next to the sea. So if you want to enjoy the south of France in September, please come. And I thank you for your attention.
Thank you very much. The talk is open for questions. I don't see any, but I would have a question. Okay. Have you tested your constructs, how they, or are they binding any other antagonist of NMDA2B receptors? So is the binding of antagonists affected or not?
This is a good question because, yes, there is interaction between this site and especially an antagonist site that is on the upper lobes of the N-terminal domains. They are mutually exclusive. But at the same time, I mean, we do not plan on using them both together because they are both used to modulate the channel in one way or the other. So I don't think that's an issue. Yeah.
My question comes from another aspect. So my group developed an GLUN1 2B PET tracer. If we would love to test it in a clean model where we just have the dimer, because otherwise it's really difficult to interpret what we get in the scans. So your mouse model would be actually perfect for that if the binding of the PET tracer is not affected. So
yeah, I mean, you will need to test. We can discuss.
Yeah.
Hello. Hi. Very nice talk, and sorry for a very, very general question. So the extrasynaptic NMDA, the subunit composition, don't have any ability to interact with the postsynaptic density proteins, or is there any difference there? Or?
Sorry, I missed part of your question because someone coughed. But,
sorry. The extrasynaptically located NMDA subunit composition, are they able to interact with postsynaptic density proteins or not?
Yes, I mean, the GLUN2B subunit is capable of interacting with the PSD95.
Okay.
So yeah.
Thank you.
There was a question on the other side.
So thanks for the great talk and happy anniversary on this project. I have a question more to the physiology of all of that. So what is the mouse phenotype? What happens if you knock out the 2A tetramer, and did you try to, I don't know, using optogenetics, induce this effect where you increase the current and see what happens in the mice?
So that's a very good question, because GLUN2A knockout mice are actually a mouse model of schizophrenia. So this would be for the next step, like can you restore some of the synaptic functions by increasing the activity of these 2B2B diheteromers in this setting, especially because we have a strong proportion of these diheteromers, and so we expect maybe to have an effect and to be able to restore synaptic plasticity, for instance.
It's a project for the next 10 years.
Yeah.
Thank you very much.
Yeah.